Today we will discuss alpha oxidation, omega oxidation, oxidation of unsaturated fatty acids, right? And disorders of beta oxidation and other disorders, right? So today I will start with the first disorder, that is SIDS. SIDS. Infant death syndrome. So the disorder is today sudden infant death syndrome. What does it indicate? The name sudden infant death syndrome. It is a disorder of beta oxidation disorder, right? The enzyme deficient is medium chain acyl CoA dehydrogenase, right? So the exact reason is not known. Exact reason is not known, right? But in 10% of the cases, that it is due to deficiency of medium chain and long chain acyl CoA dehydrogenase, right? So if this is deficiency of this enzyme is there, what will it cause problem? It is involved in proper beta oxidation, right? So if this enzyme is not present, the beta oxidation is blocked. Clear? What is the principal source of energy to the bodies? First is glucose, right? When you need energy, the body will prefer first of all glucose, right? If glucose is finished, body will break down glycogen to form glucose, right? After that, the body will use fats, right? The fats will undergo breakdown via beta oxidation, right? Now disorder is sudden infant death syndrome, right? A baby is born, mother is feeding the baby, right? Suppose the mother fed the baby last at 10 p.m. The last feed was given to the baby at 10 p.m., right? After that, the energy will be first provided by glucose, right? But after that, 3, 4, 5 hours, what will play the role of energy? Beta oxidation. But beta oxidation is blocked due to non-presence or absence of this enzyme, right? So the baby will die. Sudden infant death syndrome. Sudden infant death syndrome causes death of the baby at midnight. The mother fed the baby, a newborn baby is born, the mother fed at 10 p.m. Last feed will be given. After that, mother will also sleep, the baby will also sleep, right? So after that, the first energy will be provided by glucose, right? Principal source of energy. When glucose is depleted, the energy is provided by beta oxidation. And beta oxidation, there is deficiency of this enzyme in this baby. Medium chain acyl CoA dehydrogenase and long chain, right? So beta oxidation will not play. And beta oxidation is stopped, so energy will not be provided to the baby, so the baby will eventually die. So this is our first disorder, sudden infant death syndrome, right? We discussed the 4 steps of beta oxidation proper. 1, 2, 3, 4. Oxidation, hydration, oxidation, and cleavage, right? And in this first step, the enzyme was antibody. So beta oxidation is not playing. It is blocked and causing death of the infant at midnight. The disorder is sudden infant death syndrome, right? Disorder is clear to all of you, or anybody left? The second disorder, anybody? Second disorder is Jamaican vomiting sickness. What does the name indicate? Jamaica. Anybody who knows this country? There is a fruit there known as unripe ackee fruit. Unripe ackee fruit. And this fruit contains toxic amino acid hypoglycine A. Right? What will happen to the patient or the person who eats this fruit? This disorder is characterized by hypoglycemia, vomiting, convulsions, and even death by eating this fruit, unripe ackee fruit, because it contains toxic amino acid hypoglycine A, and it causes blockage in beta oxidation. Right. This is second disorder, Jamaican vomiting sickness. The symptoms are hypoglycemia, vomiting, convulsions, and even death. It is caused by eating unripe ackee fruit that contains toxic amino acid, that is hypoglycine A. Right? Anything left in this disorder? And it inhibits the enzyme acyl-CoA dehydrogenase. Coming on to the third disorder, methyl malonic acidemia. In the previous class, we discussed oxidation of first even chain fatty acid. Hope you remember if you have studied even chain fatty acid. What was the end product? 2 carbon. What was 2 carbon? Acetyl-CoA and acyl-CoA, right? Acyl-CoA was recycled for 7 cycles and acetyl-CoA enters the TCA cycle, right? But in odd chain fatty acids, odd chain, what was the end product? Propionyl-CoA. I told how is it metabolized? Propionyl-CoA is first carboxylated to form dimethyl malonyl-CoA. Then it is converted to L-methyl malonyl-CoA, right? Then by B12 it is converted to succinyl-CoA, and this succinyl-CoA enters the TCA cycle, right? And this is oxidation of odd chain fatty acid. Odd means they are multiplied by 3. So end product will be 3 carbon, propionyl-CoA, right? In even chain, they are multiples of 2, 2, 4, 8. The end product will be 4 carbon, and 4 carbon will undergo cleavage to form acyl-CoA and acetyl-CoA, right? Acetyl-CoA will enter the TCA cycle directly, right? But if you have odd chain number of fatty acids, right? In odd chain, the end product will be 3 carbon propionyl-CoA, right? And propionyl-CoA first undergo carboxylation reaction with carbon dioxide to form dimethyl malonyl-CoA. Then it is converted by Jasmeet reaction to form L-methyl malonyl-CoA. Then via B12 it is converted to succinyl-CoA. But if there is deficiency of B12, this reaction is blocked. So this L-methyl malonyl-CoA will accumulate and it is excreted into urine, and its level will also increase in blood. So disorder was methyl malonic acidemia. Right. It comes under odd chain fatty acids. Odd chain. In odd chains, the end product is 3 carbon propionyl-CoA, and propionyl-CoA will be metabolized via first carboxylation reaction to form dimethyl malonyl-CoA. Then D is converted to L isomer by a Jasmeet reaction. Then finally isomerase will act to form succinyl-CoA. Succinyl-CoA will enter TCA cycle and is metabolized. But there is deficiency of vitamin B12. Dr. Karan Pal also discussed in B12 as well. Sarvar Adil, you know right? So B12 is not there, the reaction is blocked, and this product will accumulate in blood, that is L-methylmalonyl CoA. So it will accumulate in blood, the level will first increase in blood, and then it will be also filtered into urine. So the disorder will come methylmalonic acidemia, right? What are the symptoms of this disorder? There are two reasons. One is due to B12 and other is due to this enzyme methylmalonyl CoA, this enzyme mutase, right? How will you remember the enzyme? MMM, methylmalonyl CoA mutase, MMM, right? So the pathway is blocked here. Either there is deficiency of vitamin B12 or due to deficiency of this enzyme. So this will not be converted to succinyl CoA, and succinyl CoA will not enter TCA cycle. This product will accumulate in blood will increase, then finally filter into urine, the disorder will come, methylmalonic acid. What are the symptoms? Anybody? So first of all, there is accumulation of methylmalonic acid in blood and urine. The symptoms are metabolic acidosis, write down metabolic acidosis, CNS damage, retarded growth, and death in early years of life, right? So now just to revise, we discussed three disorders. First disorder was SIDS, sudden infant death syndrome, right? It is due to blockage in beta oxidation. The primary source was the baby was fed on mother feed, right? The milk provides energy to the baby, but after that the second source is fats. The fats will provide energy via beta oxidation, but there is deficiency of enzyme or beta oxidation. Which enzyme? Medium chain and long chain acyl CoA dehydrogenase, right? So beta oxidation will not play. So it will not provide energy to the baby, and there is sudden death of the baby at midnight. So the name of disorder is sudden infant death syndrome. Pediatrics में भी आएगा पक्का, ठीक है? Second disorder कौन सा था? Jamaican vomiting sickness. Jamaica है, IK fruit है, that causes toxicity via hypoglycemia A. It causes blockage in beta oxidation. So the symptoms will be convulsions, coma, and finally death, right? Third disorder is methylmalonic acidemia. Remember the name, and it will come under OR chain fatty acids. OR chain fatty acid, the product, final product is propionyl CoA, and it is metabolized via dimethylmalonyl CoA L, and this reaction requires B12 and MMM, methylmalonyl CoA mutase. So there will be deficiency of this enzyme or B12. This product will accumulate, causing increased levels in blood and finally filter into urine. So the symptoms will be mental retardation is there, CNS damage is there, and metabolic acidosis, right? Now we discussed saturated fatty acid earlier, right? Oxidation of even chain fatty acids, proper beta oxidation. Then if they are odd chain, this method. Now we'll discuss which contain double bond. The fatty acid which contain double bond. Anybody who can name the fatty acid which contain double bond? Anybody? Yes. How they will undergo oxidation? Tell me, anybody? Normally there are even chain fatty acids, right? Even chain, the product was acyl CoA and acetyl CoA. After four steps was oxidation, hydration, then again oxidation, and then cleavage. We told the mnemonic OHOC, right? So even chain fatty acid end product is after these four steps, acyl CoA and acetyl CoA. Acetyl CoA is 2 carbon because it is even chain, right? So final product will be acetyl CoA and it will enter TCA cycle, right? And if these are odd chain fatty acids, then the same steps will occur, but end product is 3 carbon propionyl CoA. And propionyl CoA via this method will undergo beta oxidation and it will enter TCA via succinyl CoA. Clear? Now we'll discuss even chain is done now, odd chain is done now. We'll discuss unsaturated fatty acid. Unsaturated fatty acid means they contain double bonds. It is clear now. You have studied in class 1 too. Unsaturated means they contain double bond. The fatty acid which contain double bond is unsaturated, and if there are multiple double bonds, it is polyunsaturated fatty acids. Do you know omega-3 and omega-6 fatty acids? Do you know how to calculate? So during unsaturated fatty acids, they contain double bonds. So how they are? Will undergo beta oxidation. Due to double bonds, there is some differences in the oxidation. First is that there is less energy produced during oxidation of double bonds, and there are two more enzymes used, isomerases and reductases. What will they cause? They will break the double bonds. After breaking the double bond, then again proper beta oxidation will occur. There are additional steps. So if you have a saturated fatty acid that contains only no double bond, right? So what will happen? It will be metabolized very fast. But if it is saturated but it contains odd number of fatty acid, then it will be metabolized bit fast due to propionyl-CoA pathway, this one. But if it contains double bonds as well, unsaturated, what will happen? It will first break down double bonds and it will provide less energy. One thing is that, and two more enzymes are used for breaking double bonds. After double bonds are broken, then this pathway will start. So this is oxidation of unsaturated fatty acid. Just simple. Two steps are added additionally. Right? Now coming on to the next. And normally beta oxidation proceed for less than 20 carbons. And if a fatty acid has more than 20 carbons, what will be the process? More than 20 carbons. How will you do that beta oxidation? Have you read about peroxisomes? Peroxisomes. They are present in prokaryotes or eukaryotes? Starting from +1. यूकैरियोट्स में होते हैं ना. So if a fatty acid has more than 20 carbons, then it is metabolized via first peroxisomes. Beta oxidation in peroxisomes. It is a bit different. How? Peroxisomes are organelles present in eukaryotic cells, and it occurs via modified process in peroxisomes. How? I told the 4 steps. And before this, the steps was carnitine shuttle. This was 3rd step, this was 2nd step, and 1st step was activation of fatty acid. Total steps are these now. Beta oxidation, first is activation, then carnitine shuttle brings the activated fatty acid into the mitochondrial matrix. This is cytosol, and this is mitochondrial matrix. These 4 steps, right? Now what will happen in peroxisomes? Anybody who has studied oxidation in peroxisomes? In this, I told the first oxidation FAD is produced, and in the second oxidation NAD is used. And here 2.5 ATP are produced, here 1.5 ATP is produced, right? But in beta oxidation in peroxisomes, the first oxidation, what will happen? FAD is produced, but it will not handle reducing equivalents to the ETC. It will not handle its reducing equivalents to the ETC to produce 1.5 ATP. But what will happen? It will, anybody, what will happen? Normally FAD via reducing equivalents will enter ETC and will produce 1.5 ATP, right? But in peroxisomes, this step is not done. So FAD will enter, it will use reaction of catalase and will produce hydrogen peroxide. How? I will show you the steps. This is our chain, methyl mannonic acid, right? We discussed it, right? Now, unsaturated fatty acid हमने कर लिए. This is peroxisome, right? This is the reaction FAD to FADH2. So what will happen? Normally FAD is converted to FADH2 and 1.5 ATP is produced, right? But in this case, FAD is handed over. This is blocked, and FAD is handed over directly to oxygen. So it will produce hydrogen peroxide, H2O2, and then it will undergo reaction via catalase to form water and oxygen, right? But energy is liberated as heat only. No ATP is produced. That is difference of peroxisomes in beta oxidation. So how will you choose a fat? Which pathway will it undergo? First thing is that if it's plain or even chain fatty acid. Even chain fatty acid is there, and end product will be 2 carbon. 2 carbon product will be acyl CoA and acetyl CoA, right? Acetyl CoA will enter TCA cycle directly, it will be metabolized. Now comes odd chain. Odd means multiple of 3. The product will be multiple of 3. The 3 carbon will be methane, ethane, propane. Propaneol CoA will be formed. 3 carbon propaneol CoA is formed. Then how will be it metabolized? Via succinyl CoA. Succinyl CoA TCA cycle, right? Now after that, these are fatty acid which doesn't contain any double bond. Now coming on to unsaturated fatty acid that contain double bond. What will happen in case of unsaturated fatty acid? The double bonds will be broken down, and then finally beta oxidation will proceed. To break down, what will you use? 2 more enzymes, isomerases and reductases. They will break the double bonds. After that, there will be no double bonds. And then there is no double bonds, proper beta oxidation will proceed. How? If there is even chain fatty acid, beta oxidation will proceed via acetyl CoA. And if it is odd chain, via propaneol CoA. Done. Now coming on to, if it has more than 20 carbons, up to 20 carbon it will undergo beta oxidation directly. But if it has more than 20 carbons, it will first undergo beta oxidation in peroxisomes in eukaryotes. What will be the step? The earlier fatty acids are broken down via this pathway FAD to H2O2, catalase and water and oxygen, right? After that, when 20 fatty acids carbons will be left, then proper beta oxidation will proceed, right? And this, right, the reducing equivalents from FADH2 are not transferred to the ETC but handed over directly to oxygen in this pathway, and FAD to oxygen and H2O2 will be produced, and via catalase will form water and half oxygen. And there is no ATP synthesized in peroxisomal, which is produced by a high fat diet and administration of hypolipidemic drugs. Now coming on to the next disorder, Zellweger syndrome. Zellweger. Other name of this disorder is cerebro-hepatorenal syndrome. Zellweger. Zellweger syndrome. What is problem in this disorder is that the beta oxidation in peroxisome is blocked. In peroxisomes is blocked. Right? So if the fatty acids are more than 20 carbons, they will not be metabolized. Right? So what will happen? They will accumulate in soft tissue. First is cerebrum, then liver, then kidney. The problems: cerebro-hepatorenal syndrome. If you are not able to remember the Zellweger name, remember it is that the large chain fatty acid that will not be metabolized via peroxisomes, they will accumulate in brain, cerebrum, hepatic liver and renal kidney. Cerebro-hepatorenal syndrome. As simple. So they will accumulate in brain, liver and kidney. The disorder is also known as cerebro-hepatorenal syndrome. Clear? Now coming on to the third, alpha oxidation. Alpha, what does alpha mean? So now we will discuss alpha oxidation. In previous class we discussed beta oxidation. What is alpha oxidation? I told you, hope you remember in the previous class, CH2, 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 COO. H. This is a normal fatty acid, alpha, beta, gamma and so on. Beta oxidation happening at beta carbon, alpha oxidation happening at alpha carbon. What was the problem? Just remember the basic things. Basic chemistry plus 1 2 में पढ़ के आए कि नहीं आए? And simple thing was that 1 carbon methane, 2 carbon ethane, 3 carbon propane, and in odd chain we formed the propionyl CoA, and even chain we form the 2 carbon acetyl CoA. What is the difficulty? Just keep on studying, na? Try until you succeed. I want you to succeed, right? So we will forming now coming on to that main topic, alpha oxidation. Mr. Dhruv, now you can remember alpha oxidation is happening at alpha carbon, beta oxidation is happening at beta carbon. We'll discuss later omega oxidation, that will happen at omega carbon. What is the difficulty, na? Right? So we start with alpha oxidation. You can see it, Mr. Dhruv. Yes. See, right, what is written? Oxidation at alpha carbon atom is known as alpha oxidation. As simple. Removal of 1 carbon unit from carboxyl end. Alpha oxidation doesn't involve binding of fatty acid to coenzyme and no energy is produced. And there are two differences in alpha oxidation than beta oxidation. Anybody? No energy is produced, right? And in beta oxidation, beta was second carbon, so 2 carbon units are used. In alpha, 1. But simple, just simple, right? When will it undergo? You have a fatty acid given in the body. How will your body decide I will go for beta oxidation or alpha oxidation? How will your body decides I will do beta oxidation or alpha oxidation? If you are taking fats, you are eating some omega-3 flax seeds, some good fats, coconut oil, right? And after that, what will your body decide? I will go through beta oxidation or alpha oxidation or omega oxidation. How will your body intelligence will decide which oxidation is going to start? You are taking any fat, right? So first process is beta, that is predominant pathway. Predominant pathway, so it will mostly occur, right? The first of all, the body intelligence will decide whether it is saturated fat or unsaturated fat, right? It will break double bond, then it will proceed to beta oxidation. See it clearly. If it has more than 20 carbons, right? First process is it will break up to 20 carbon via peroxisomes, beta oxidation, peroxisome. After that, 20 carbons will be left. And after 20 carbons, it will see it is even chain or odd chain. If it is even chain, it will form acetyl CoA and beta oxidation, propionyl CoA and beta oxidation. But before that, if it contains double bonds are there, then it will first break double bonds and then beta oxidation will proceed. Yes, great intelligence. You are eating any fat, but body knows I will go beta oxidation, I will go mega oxidation, I will break through peroxisomes, I will have even chain fatty acid, I have odd chain fatty, it will break down. You even don't know, body knows, right? So now alpha oxidation I told, it is oxidation at alpha carbon, right? And beta oxidation beta carbon. Beta is second carbon, so 2 carbon fragment is first released, and in alpha 1 carbon is released. Just simple. Anything left? And in alpha oxidation no energy is produced, right? Moreover, next disorder is Refsum disease. Refsum. You will get minimum 4 or 5 questions out of this. What is Refsum disease? It is a rare neurological disorder characterized by peripheral neuropathy, and cerebral ataxia. Peripheral neuropathy is there. Cerebral ataxia. There is accumulation of phytanic acid. Phytanic acid in body. Let's move back to +1 +2. Did you did the structure of chlorophyll? Chlorophyll की structure की थी +1 2 में? Chlorophyll. Hope you remember chlorophyll contains a ring at the center that is known as phytol ring. Anybody who knows, just raise hands. Backbenchers. Phytol ring याद है किसी को? क्लोरोफिल में phytol ring, phytanic acid, this disease. So there is deficiency of phytanic acid alpha oxidase. In phytanic acid, there is presence of methyl group at carbon 3 here. So it will not undergo beta oxidation proper. First, it will remove 1 carbon as carbon dioxide followed by beta oxidation, right? Derived from phytol ring of chlorophyll, and it's mostly found in plants, right? And phytanic acid accumulates. Enzyme deficient is phytanic acid alpha oxidase, and it will cannot be converted to finally get by beta. So how will you treat this disorder? How will you treat this disorder, or just relieve the symptoms of this disorder? We discussed the thing coming like phenylketonuria, anything else. If you are not eating phenylalanine, how will the disorder appear? If there is no enzyme deficiency, no problem. But if you are not eating phenylalanine, the disorder will bit decrease. In the same manner, this is problem from green vegetables or green foods, because it contain chlorophyll. If you stop eating chlorophyll diet, problem will come? No, or it will decrease the symptoms of disorder, right? Because the root cause of problem is phytol ring. Phytol ring is all present in green color plants, right? So because it is not undergoing oxidation, the symptoms of peripheral neuropathy are there and cerebral ataxia is there, right? So how many disorders we discussed today? First was, one was Zellweger syndrome, one was Refsum disease, then previous Jamaican vomiting sickness, sudden infant death syndrome, right? Now anything left? One oxidation, omega oxidation. Omega oxidation. What is omega? Omega oxidation. Now you can tell me omega oxidation. Beta oxidation at beta carbon, alpha oxidation alpha carbon, omega at omega carbon. It is a minor pathway, right? Right? And it involves hydroxylation followed by oxidation at omega carbon. There are two steps. First is hydroxylation and then oxidation at omega carbon. Just simple. Just simple. Now you can, if you, this question will 100% come either in 5 marks or 10 marks. My promise. If I will have a percent, I will add it. Okay? And second thing, if you are not able to remember the 4 major things that first during beta oxidation, first is activation of fatty acid, right? Then activated of acid is activated in the form of acyl CoA that is not permeable into the matrix where proper beta oxidation is going on, right? It is carried inside via carnitine shuttle. Then it is proper beta oxidation. We have 4 steps: O, H, O, and C. Oxidation, hydration, oxidation, and cleavage, right? And finally, we will form acyl CoA and acetyl CoA. Acetyl CoA will enter TCA cycle because it is we are going beta oxidation in which even chain. Even means multiple of 2, and if it is odd chain, multiple of 3, we will get the end product propionyl CoA 3 carbon. Methane, ethane, propane is 3 carbon, propionyl CoA. Propionyl CoA will form dimethylmalonyl CoA, L-methyl, and finally succinyl CoA. Then it will enter TCA cycle. It is done. Now the fatty acid is having double bonds. What is the problem? We will break double bonds first, then will it undergo beta oxidation. How will you break double bonds? You use 2 additional enzymes. One is isomerase and second is reductase. They will break the double bonds and finally it will undergo beta oxidation, right? It is done now. Even chain is done, odd chain is done, and double bonds unsaturated is done. After that, we use, if a fatty acid is having more than 20 carbons, what will you use? Beta oxidation peroxisomes, right? You will break down first 20 carbons greater than 20, then you proceed via beta oxidation, right? And there are 2 minor pathways, one is alpha and one is omega. Omega, the first step is hydroxylation and the second step you use oxidation and omega carbon, right? But in alpha oxidation, what's the difference? Oxidation at alpha carbon, but any difference more? Anybody? And there is no activation required here in alpha oxidation. Directly काम शुरू हो जाता है. And one more thing left, metabolic water. Metabolic water. What is metabolic water? Metabolic water. Have you seen camel? Have you seen kangaroo rat? Camel has a hump, you know. It lives without water and food for so many days. How? The hump in the camel has fat, and fat is broken down to form water and energy. By this mechanism, the camel survives via metabolic water. And kangaroo rat doesn't eat anything, but it eats oil-rich seeds. The seeds will undergo beta oxidation, provide energy and water to the animal. Anybody who has doubts, please tell me. This is production of water. 1 molecule of palmitic acid will undergo beta oxidation to produce 16 molecules of water. And application is in camel hump. In deserts, there is lot of no water, no food. The camel survives via beta. So camel hump will provide energy via beta oxidation, and this kangaroo rat as well uses oil-rich seeds for its survival. Clear? Thank you. That's all over.